Hi friends, welcome to day 2 of answer writing program. Discussion for today. Question is examine gene therapy and write a short notes on mitochondrial replacement therapy. So first thing what we should do, identify how many parts are there in the question. So this is first part, and the first part is explain the gene therapy, and the second part is write a short notes on the mitochondrial replacement therapy. So I have identified there are two parts. Then what is the second step? Identifying the question tags, examine, and also write a short notes. Now based on the yesterday's discussion, can you guys pause and think how much weightage should I give for two parts in the answer? So what would be the weightage for the first part and what would be the weightage for the second part? Pause and think for a while. Okay. Now the answer is I will write 60 to 70 percentage of this part and rest of the 30 to 40 percentage for this part. Why? Because here the keyword question tag is examine, and here it is simply given write a short notes. I hope this part is clear. This is a very important part, guys. This is very important to bring these things in the exam, be it for any exam. Does not matter UPSC, TSPSC, APPSC, or does not matter any PSC. These are the basic things that you should keep in mind. Now we are discussing gene therapy, very important for this year based on the questions that were asked before. So gene therapy, now let's understand the concept. Like yesterday I have told you, we have the cell. Within the cell there is a nucleus. Within the nucleus you have the chromosomes. On the chromosomes you have the DNA. Let's assume the DNA, and within this DNA there are various part. On each part coding is written, just like a software code it is written, and this coding is for every information that our body has to do perform, like your body color, your organ development, your eye color, hair color, anything you take, it is based on this coding. Okay, and this is called gene. So everything happening because of this coding. Now what if this coding is defective? For suppose you have sickle cell anemia. In this case, what is happening? The red blood cells are defective. These red blood cells, these play critical role in development of or transportation of the oxygen and all these things. So when this thing is defective, you will feel anemic. And you might have also heard about the problem of blood clotting, that is called hemophilia. Okay, you might have heard about this thing, blood clotting, whatever. Let's say some injury has happened. In that case, whenever the injury happens, your wound should heal. The blood should stop pouring out. That is called blood clotting. In some cases, that will not stop, and that genetic. These are the certain genetic disorders. Okay, so these are some faulty genes that are there. Now, gene therapy is trying to correct this faulty gene. So you have to either replace this gene or just correct this gene in order to resolve these genetic disorders. Sometimes these genetic disorders are also transferred from parent to its baby, their baby. Okay, so even these things can be corrected. So this is a very important technology. I hope the concept is clear. So gene therapy is a technique that modifies a person's gene to treat or cure disease. So it is a collection of methods. There are various methods that allows correction of the gene defect that has been diagnosed in the child or the embryo. Now, gene therapy, what are the mechanisms? So you are replacing a diseased gene with a healthy one, like the thing I have mentioned. See, the diagram here shown, it is a healthy gene. Yesterday we have discussed recombinant DNA, right? So we are combining two genes, right? Two different genes, foreign gene and the host gene. Now this is a healthy gene. How will you transfer this healthy gene into the human being? You have to use some kind of vector. This is the thing that we have studied in the recombinant DNA. You will use the vector, you will do some kind of cutting, you will transfer this thing in the vector, and then finally you will transfer into the human being. Yesterday we have discussed this thing, process of rDNA. So in this way, healthy gene is transferred, or else some gene, because of the gene expression, that is causing the problem. So you can inactivate a disease causing gene, or you can modify the existing gene. Simply there is a gene, you have to cut and you can simply modify this gene. Okay, these are the various ways that gene therapy can work. Now, what are the different types that are there? All this is examination. You are doing a deeper examination. Okay. So when you transfer or modify genetic structure in yourself, let's say I have, why to take you? I have some kind of cancer. So if I do modification in my body in a cell called somatic cell, somatic cell. There are two types of the cells in a body, primarily somatic cell and the germline cell. Okay. These germline cells are responsible for the reproduction and they do not play in the reproduction. So if I make any kind of modification in the somatic cells, whatever the genetic modification will happen only to me. If I make modifications in the germline cells, such a modification will be transferred to my next generation, my children or anyone. Okay, so these are the types of somatic cell gene therapy. And also, let me tell you guys, CRISPR-Cas9, you also need to study. This is also one of the way of gene editing. This is a tool of gene editing, very important, CRISPR-Cas9. I think already APPSC has asked. So that is one of the way for cutting this way. So here only difference is just like rDNA, where you use the restriction enzymes. There you, like I have posted in the Telegram also, there you will use protein for cutting up of the DNA. Here, instead of protein for cutting, here you actually use Cas9, but location of this particular aspect where you have to cut, it is done by RNA, mRNA. You will do this programming. Okay, you guys need to study this CRISPR-Cas9 also. Okay, let's come back to our discussion. Advantages of promoting the gene therapy. Okay, so permanent result may be possibility. We have high burden of the rare genetic diseases like the things I have mentioned, like sickle cell anemia or hemophilia. All these are so many problems that India especially is facing. And this can also be a kind of medical tourism. It can be promoted. Okay, worldwide there are so many such kind of genetic problems. If India can develop these gene therapies, and it can be also a good market. Firstly, the health disease burden, especially for the people of the India, this can be effective treatment. So it can also helps fulfilling the DPSP, okay, social justice role of the government. So these are the certain advantages. Like yesterday, I told you how to generate the points. You always think from the stakeholder point of view, from the government, from the people, okay, normal people, for the business people. Okay. So in this way, you will be able to generate more points. For the government, it is doing this role of social justice. For the business people, you are able to generate more kind of revenue, right? For the people, treatment can become affordable if it is developed in India. So their health problems can be resolved. So we will be able to get better health. But what are the challenges? Sometimes, so these all this you have this. If the question comes on the gene therapy, you have to just pour out the 7 points. You cannot just go and brainstorm there. So it should be ensured that only undesired gene is properly treated. Okay, if you go and do affect some kind of gene, like say this is the faulty gene that you have, you should not affect the other genes. Okay, that collateral effect should not be there. Properly target this thing. So CRISPR-Cas9 is one such technology is able to effectively target at the place where it wants. So that's why I said CRISPR-Cas9 can be important question. And sometimes, even let's say I have added a gene, I'm facing some kind of cancer, and I have added a gene from the healthy person. But sometimes my immune system can reject that healthy gene. And now I've told you I will use the virus as a vector for integration into the host. But this can sometimes show the toxicity, delivering it to the right place. And this is a high cost, even though if invention is done in India, so all the research is done. So still it is highly costly and not affordable to every person. So this can again cause problems to the poor people. Okay. And what if, like say, X-Men kind of situation, okay, genetic mutation, not just mutation kind of thing, and they wanted to do some kind of war. Okay, so just genetically manipulation, let's I have done some kind of changes in the germline germline cells. So what happens next, even in the next generations also, babies will take birth with the modified genes. So this can be a possibility of biowar. Okay, China has done, okay, most of the Indians, they have changed the gene of the parents. Now they will reproduce. So automatically, slowly, it will be transferred to different people, right? So there is such kind of cases. So it should be very careful. And sometimes it cannot correct problems like Down's syndrome. Down's syndrome is a chromosomal problem. Now, chromosome, it has many genes. So if you have problem in some or one gene, it can be corrected. In the Down syndrome, it is problem of chromosomes. So chromosome means having set of genes. So you cannot correct the chromosomal disorders. That is also one of the important point, guys. Okay, all these are the very important points that you should take it to the exam hall. Then what should be my conclusion here? ICMR has given certain guidelines which should be implemented to ensure ethical, scientific, and regulatory requirements for developing and conducting clinical trials on gene therapy in India. That should be properly implemented. So what are these detailed guidelines? I have given it in the additional information, guys. You can read it. Okay, I have given this thing. But for the answer, this much is enough. So we have answered the first part. We have given detailed examination. So what kind of mechanisms are there? How many types are there? And then advantages. You can you in the challenges, the ethical part also you can add. In the advantages, you can always use your own innovation to come up with a more number of points. We have discussed the first point. Now come to the second point. Mitochondrial replacement therapy. Now, in this case, okay, we have, so let's discuss this thing. Okay, this is the cell and we have the nucleus. Now, generally, okay, how the fertilization happens, reproduction happens, there is a sperm cell, okay, sorry for my diagram, and there is a egg cell, okay. When these two cells, mother cell and father cell combines, okay, finally a zygote will be formed like this, okay, within the egg cell itself, okay, and there would be half nucleus will be coming from the mother and half nucleus will be coming from the father like this. Okay, so okay, not this thing. Okay, okay, fine. Okay, there would be let me see. So there would be two nucleus like this. Okay, over a period of time, this will become a single nucleus like this. Like this. Okay, this half is coming from the mother and half is coming from the father. So nucleus developmentation. In this nucleus, you have the chromosomes, DNA, right? And the rest of the part, here you have the mitochondria. Mitochondria has also DNA, though it is a little part of the information. So there is a mitochondria. Now, who is providing the mitochondria? Mother is providing the mitochondria. Okay. So only the sperm cells nucleus will be going into the mother cell and then finally forming. Now, this rest of the part of the egg cell, it is same. In this nucleus, father's nucleus will also enter. So this egg cell will accept like this two parts. It will accept, right? And rest of these parts will be same. There is a mitochondria and this fertilization will happen. Now, this part, this mitochondria is defective. So we have to make sure this mitochondria is replaced. How would I do? Now the fertilization has happened. So this is the faulty part, right? This whole part on where DNA is coming. Okay, the genetic material is coming from the mother and father. Okay, so I will take this part and I will take the healthy egg cell of the donor and I will place this nucleus in the donor cell. Okay, then finally, for development of the cell will happen and it will again grow back in the mother. Okay, so donor here is contributing to the egg cell. After total development of the complete cell, then again it is transferred back to the mother. So this is one way. There is also second way. This is the pro-nuclear transfer. This is being done after fertilization, right? After fertilization, fertilization means mother cell and father cell combining to fuse together, forming of the zygote. Then I am removing this nucleus and putting this nucleus in the healthy egg cell of the donor. This is happening after the fertilization. I can also do this thing before fertilization. In the before fertilization itself, I am taking this mother's nucleus and father's nucleus and I am putting it in the donor's egg cell and there I'm doing the fertilization process, the fusing process. Then this complete cell will be again transferred back to the mother. Okay, so this is the thing that is happening before the fertilization process. These two processes are important, friends. People know only one process. This is very important this year. Now see why this thing is called a three-parent baby. So every time we consider, why do we consider our mother and father as a parents? Because we derive genetic information from them. But genetic information comes from mitochondria also. Okay, but here mitochondria is coming from the donor. So because of this thing, genetic information is coming from the three parents. We call this as a kind of three-parent baby. And this therapy is called the mitochondrial replacement therapy. If you don't do this mitochondrial replacement therapy, if your mitochondria is problematic, it is going to cause severe problems to the child. So there are two methods. First is pro-nuclear transfer after the fusion. That is what it is given here. Then second is a reconstructed egg technology. That is before the fertilization process. Okay, I have given the steps here. Remove the nucleus of an egg from an egg donor who does not have the mutation, replacing the nucleus of the egg with the nuclear DNA from the woman who has mitochondrial DNA mutation. Fertilize the egg with the father's sperm in the embryology lab. Okay, simple thing. So the same thing it has been mentioned. Recently, last year, the UK has gave the approval for this mitochondrial replacement therapy. So this is becomes very important for this year's exam. So two process I have given, I have mentioned things here, pro-nuclear transfer. What is that is happening here? It was it is a reproductive technique that involves transferring of the nuclear DNA of a fertilized egg in a donor egg that has its own nucleus removed. So simply from the cell, remove the nucleus. Okay, keep it in a this nucleus in a egg cell which is healthy, which does not have any problem. Because why? In this case, from the zygote, if you allow this thing only to fertilize, here you have the faulty mitochondria and this can cause a problem. Here in this case, donor will provide this mitochondria and this nucleus will be uh, implanted into this donor, and again after development, again it will implant it to the mother. So this explanation is given here. Now, what is the significance of mitigating the mitochondrial diseases, prevention? Now, hope for the families. Instead of fearing that this problem can cause to the children, I will go childlessness. So, if there are genetic disorders, like I have the, if my wife have the mitochondrial disorder, so she might be afraid that she will prefer being childlessness because it can affect to the her child. Okay, so because of this treatment, this childlessness can be eliminated. Okay, she can bear the child. Then there is a scientific advancement. By doing this more research, science will be advanced. Now what are the issues? Obviously this ethical controversy comes in everywhere in the biotechnology, because you are playing with the act of nature, genetic changes you are doing, and you do not know the long term impacts that is going to have. And this process is not going to be simple, complex process. These points you can write it everywhere, and again this is not affordable to everyone. This can also be written, you can also write here also, social justice thing can also be written here. How many questions there will be? Four or five questions will come from the science and technology. This applies to everywhere. You have those set of points that can be used to substantiation everywhere. Medical research and development can improve, tourism can improve, all these things can be incorporated everywhere. Okay, so again, what should be my conclusion? So every time when I talk about the problems, then I have to give you some kind of futuristic conclusion. Invest in research to understand the mitochondrial replacement, there is the implications better. Develop clear guidelines for its clinical use. Ensure that MRT remains accessible to those who need it. Clear, guys? So though I have given equal weightage, like have explained all the things in detail, but you have to make sure while writing that this part you have to write in a short form in this part, because you cannot write everything like based on the things I have discussed to the toppers. It is better to write around 2 to 2.5 pages. Okay, something at the max 3 pages. Anything, it's not advisable to write in 1 page or 4 page. If you write more than this, you have to compromise quality somewhere. If you are writing somewhere like this, 2 to 3 pages, okay, you will be able to make sure that quality is good. Okay. I have given the additional information. Now, one more point from my side. I have added PYQ here. The reason is few of the people have the doubts. They are preparing for the group 2 as well as the group 1 or as well as a UPSC. I have asked them to do the integrated preparation. Now, how do I do the integrated preparation? I have studied the concept of this gene therapy, or this thing, mitochondrial therapy and gene therapy. Now, what are the PYQs that were asked from this question? I'll try to solve them. See here, these were the PYQs question. Now, please go through this. A pro-nuclear transfer that was asked already. This was also asked already. Please try solving them. Now, in this way, when you solve the PYQs, it will clear your group 2 and the UPSC. Okay, while what is the difference between group 2 and the UPSC? They will ask more factual, but at the same time, conceptual clarity is also important. So it is better you can solve the UPSC PYQs. With the solving this UPSC PYQ prelims, then you are integrating the UPSC prelims as well as the group 1 mains, and the same way for the group 2, but you have to focus on little bit more factual information, like they asked in the group 1, which institution is involved in this developing this IIT, IIM, which shipguard like Cochin shipguard is developed this hydrogen fuels. So like this, they are asking factual things. So those also you need to focus. But always you have to prioritize one exam. You cannot prioritize all the exams equally. If you are doing UPSC and group 1, prioritize one exam. If you are doing UPSC and group 2 or group 1 and group 2, prioritize one exam, but however, attend both the exams. Okay, guys. I hope the discussion is crystal clear. And like I said, only discussion can also be enrolled. Only there you will get the synopsis along with the discussion videos, both for the answer writing and the essay. If you have any concerns and any queries, please follow me, ask me on the Telegram. Okay, guys, bye bye. Take care. Thank you. Study well.